Now, admittedly, CFTR mutation assays aren't new. Universal CF screening was advocated in the US in 2001, and a number of different kits are available in the market. But these kits are only as good as the mutations they target, and content drives the coverage to detect pathogenic variants in CF patients and in carriers. So one of the most powerful resources for identifying and characterizing CF mutations is the CFTR2 database. The database compiles clinical, functional, and genetic data from about 90,000 CF patients and includes more than 350 CF-causing variants. But a limitation of this database is that patients in it are 95% Caucasian, and thus its composition is decidedly homogeneous. Now, despite this limitation, it has been a primary resource for selecting variants for targeted CFTR mutation assays. And as such, its use is similarly constrained in that it does not represent the genomic diversity seen in many populations, such as the melting pot in the US. And further, for purposes of carrier screening, it presumes that the prevalence of carriers in a non-CF population can be inferred from variant frequencies in CF patients. Now, recently, this gap in genetic diversity was addressed in a study of 115,000 individuals from the US who had their CFTR gene sequenced. This cohort, unlike the composition of CFTR2, was representative of the eclectic US population. So about 60% were Caucasian, and relatively large portions of the study group were African American, Hispanic, or Asian. A total of 213 CFTR pathogenic variants were identified in the study, and combined with other resources, these data can help guide panel design to make CFTR mutation assays more inclusive and more ethnically diverse. Data can also help us build assays that focus on what matters most, and that is coverage. By that I mean identifying more individuals with disease-causing variants in a real-world population, rather than fueling an arms race of more variants for the sake of having more variants in a panel. So let me quantify this position by showing you how these data may be leveraged to help improve the coverage of those with pathogenic CF variants. So this plot is showing the percent of the US population on the y-axis that would have a pathogenic or likely pathogenic CFTR mutation against currently available commercial targeted panels using the data from the recent study. The x-axis shows the number of variants that each kit reports. And as you can see, none of the kits offers more than about 88% coverage of CFTR mutations in the US demographic, not even those with up to 139 variants targeted. Now, in contrast, our panel content is informed by the most recent and comprehensive data for this population, and thus provides a relatively large bump in comparative coverage to 93% of the population. This analysis shows then a clear improvement in coverage for the diverse US population. But what would it look like if we used the CFTR2 database instead? Well, coverage is almost the same at 92.5%, and it's still better than all other non-NGS targeted kits. That's a strong value proposition for coverage alone, using the Asuragen kit as designed. But the devil's in the details, and the benefit of a targeted kit is that it provides ready-made classification of curated content with the assumption of a FACIL workflow, something that's far easier to run and interpret and support than whole gene sequencing. So what can we say about that for the kit that we're developing? Well, we designed the kit with simple operation and a streamlined workflow using the same PCR capillary electrophoresis approach that we've developed for our other AmplideX kits, such as those for Fragile X and for SMA Plus that we'll hear more about today. The overall approach for the CFTR mutation assay is to use highly multiplexed allele-specific PCR. So each locus, wild type or mutant, is targeted by a highly specific primer that creates a distinct amplicon that's dye-tagged and then migrates on CE with a mobility that uniquely flags each variant. This means that SNVs and indels and repeats such as poly-T, poly-TG, and even copy number variants like exon 2,3 deletions can be detected and their zygosity can be determined. Now, make no mistake, the PCR is complex, but importantly, we design our assays so that the complexity sits behind the user in the reagents that we make in QC and not in front of them in the workflow in the lab. And as a result, the entire set of mutations for CFTR that provide the 93% coverage that I've noted can be determined through an assay that only takes about an hour of hands-on time and a total turnaround time of about 5 hours from DNA to genotype. So here's an example of the raw output from the CE electropherogram using this prototype assay. And in this case, the wild type alleles are collected in one dyed channel and the mutant alleles in a second channel, and the poly-T-TG repeats are in a third channel at the bottom. In this example, nearly all the amplicons that are produced and the peaks associated with them are wild type. But this is a CF positive sample, and you can see the delta F508 mutation is shown here in the mutant channel, and an R117H mutation shown here. And this is consistent with the reference samples annotation, including the 5T12TG annotation for IVS8. In addition, we incorporate SDRs, as shown here, which fingerprint the sample and can help avoiding mix-ups across samples from one tube to the next. So that's what the data looks like. The next question is, okay, how does it perform? Again, I'll note that this assay is still in development, but nevertheless, we've generated meaningful data from both cell lines and residual clinical samples. So to date, we've analyzed 59 mutant cell lines, representing a total of 65 het calls and 5 homozygous calls. And a total of 44 different mutations are represented in that cell line collection. All mutations and their zygosity were correctly called, and the poly-T and poly-TG status was also correctly called from those cell lines. And for clinical samples, we recently worked with a clinical lab that has significant experience in CF mutation testing. And most recently, they provided 74 samples, representing an overlapping set of variants reported by our prototype assay and their reference assay. And those 74 samples included 21 unique mutations and were distributed as 21 samples without any CFTR mutations, 18 that were homozygous or compound heterozygous mutations, and 35 that were simple heterozygous mutations. And as you can see here, all of the mutations, all of the samples were correctly called with the prototype assay. So I'll wrap up here with one final point about coverage. We can talk about more and more variants, but what matters is detecting CF patients and, where appropriate, carriers. In the US demographic, I described how the kit design and content can provide about a 5 to 7% better coverage in principle than the next best targeted kits. And I want to show you one other way to think about what a 5 to 7% difference means. So let's assume 100% analytical detection for each panel, and that each variant in each panel is fully pathogenic as manifest in a carrier couple. In this case, if 93% coverage that would be provided by our design would detect 20 at-risk couples, then a loss of 5 to 7% coverage for any individual carrier would translate to missing 2 to 3 at-risk couples by comparison. And the minimum 23 variants that are recommended by ACMG would equate to missing 5 of 20 at-risk couples. So again, coverage is what matters, not just the simple number of variants. And we believe that we have the best data-driven panel design in a simple, easy-to-use kit format that can provide better solutions for labs and for patients. So at this point, I'd like to introduce our next speaker, Dr. Henny Limmick. Dr. Limmick received his PhD in genetics from Radboud University Medical Center, and since 2000 has worked as a clinical laboratory geneticist in the genetics department of the University Medical Center, Groningen, with special interests in genetics, neurological and neuromuscular disorders, and skin disorders. Of note, the Department of Genetics in Groningen was one of the first labs to introduce a quantitative technique to measure SMN1 gene copy numbers. And Dr. Limmick has continued that tradition by closely working together with colleagues for molecular genetics research and in the clinic, and as an SMA scheme organizer of the EMQN to assess reports from many labs from around the world.